Good afternoon, everybody. It's my pleasure to introduce today's speaker, Dr. Frank Zhang. He's currently a postdoctoral fellow with the title of Flatiron Research Fellow in Olga Chuan's Kaya's group at the Simons Foundation. I think he works at the Flatiron building in New York City. Frank has previously received a Bachelor of Science degree in Biological Sciences from the Zhejiang University. After that, he came to the US. At UCLA, he received a master's degree in statistics, followed by a PhD in bioinformatics. And then now he's pursuing postdoctoral training. He's worked on initially machine learning and statistical method development for prediction, interpretation of human molecular genomics data. And more recently, he's expanded into more cutting-edge neural architectural machine learning methods for automated application of deep neural network in genomic data. And today, the title of his talk is Deciphering Human Disease by Big Data Analytics and Automated Machine Learning. We'll let him start, and then at the end, he's either submit your question in the chat or unmute yourself, and then we'll go from there, okay? Frank, you can take it away.
Okay, yeah, thanks, Jimmy, for the introduction. Let me share screen first. Okay, I assume you can all see my presenter view. Yeah. And yeah, I'm really happy to be here virtually to present my work on how I leverage big data analytics and automated deep learning to decipher human diseases. So as a computational biologist, my research work is on the intersection of deep learning, statistics, and biological data analysis. So there's little doubt that today's biomedical research has entered this era of big data. However, the conversion of those big heterogeneous data into biological knowledge and insights is still challenging. In general, my overarching goal is to accelerate this process with interpretable and unbiased modeling approaches. And my current and past research spanned several different fields, including deep learning method development for regulatory genomics, gene regulation on both the transcriptional and post-transcriptional level, and how I use the CNN to model genomic sequences and predict variant effects. So for the, more particularly for the deep learning method, I'm really excited in exploring those automated deep learning methods recently. So this refers to algorithmically designed deep neural network architectures with little or no human interventions. And prior to that, I've been developing methods of deep learning to analyze RNA alternative splicing. On the gene regulation of post-transcriptional and transcriptional level, I've been working on methods to analyze protein-RNA interactions, RNA modification sites during my PhD. And more recently, which I'll also talk more about it today, is how we analyze the immune response to SARS-CoV-2 infection. And finally, we can leverage those convolutional neural networks to model genomic sequences and interpret the genetic variants. And I do this on both the bulk tissue level and on single cell level. So as you can see here, my research really spans both novel computational method development as well as biological data analysis. And I think that's important. That's an important interaction to have to enable this better analysis through novel computational method development. And also the experience of data analysis will again inform the next generation of method development. And today I will mainly talk about two projects. The first one is about SARS-CoV-2 infection on the transcriptional and post-transcriptional level. And the second is how I leverage automated deep learning to interpret variations of genomic and molecular profilings. And since, as Brian mentioned earlier, this is a like hybrid recruiting event, I'll wrap this up with outlooks and future directions from my research. So without further ado, I'll start with the first story of today, which is how we try to understand the sex differences in SARS-CoV-2 infection. So the COVID-19 pandemic has been there for around 2 years, and that's the main reason that we're still doing this hybrid virtual event. And across the globe, COVID-19 has caused numerous deaths. And if you look across different countries, male sex is a significant risk factor with a lot more male patients ending up in ICUs or died due to complications of COVID-19. And the basis of these sex differences has been explored for therapeutic implications. So for example, several prostate cancer drugs have been repurposed as an attempt to treat COVID-19 patients. However, the study of sex difference is highly non-trivial, mainly because the presence of confounding factors. So on a molecular level, we know that males and females have very different hormone responses, and that exists before the infection and may also play a role during the infection of fighting against the virus. And aside from the obvious molecular differences, males and females also tend to have different social and environmental effects. They also tend to have different comorbidity ratios. And all those factors together makes the study of sex differences in response to COVID-19 highly challenging. So to answer this question, we leverage a unique cohort called COVID-19 Health Action Response for Marines, or CHARM in short. So the CHARM study followed more than 3,000 US Marine recruits longitudinally with weekly symptoms screening, PCR testing, and antibody titering. More importantly, this is a big collaboration between the US Navy labs, several clinicians at the Mount Sinai, and several research groups across multi-institutes. And we have the molecular profilings that exist before they're infected, they're healthy controls at the time that they're first-time PCR positive, all the subsequent PCR positive, and after they recover from this infection. Because they are Marine recruits, they're receiving this military training and are living in the same environment, and that they are young and healthy adults without strong comorbidities. All those factors taken together, this really makes the CHARM cohort an excellent resource to study how sex plays a role in the response to COVID-19. So we first started by looking at the symptoms. So a large proportion of our cohort is asymptomatic, meaning that they have a positive PCR test from their nasal swabs, but they don't experience any of those COVID-19 symptoms. But if you look at the ratio of asymptomatic across different sexes, they actually differ significantly. In particular, females are significantly higher to have a symptom due to the virus infection. And this is the case across 13 different symptoms that we recorded. And for each symptom, females are always more likely to report a symptom. And this is also true for fever, which is measured instead of self-reported, indicating that this is not simply due to a reporting bias. At the same time, we can also measure the viral load measured by CT values. So here, note that higher CT values will correspond to a lower viral load. And we can see that females actually have a lower viral load compared to the males, despite the fact they have more symptoms. So there's seemingly contradictory clinical outcomes really motivates us to study the underlying molecular differences that contribute to these differential clinical outcomes. So that we analyzed both differential gene expression and differential alternative splicing using this cohort separately within males and within females, and we were able to identify a large number of sex-specific molecular signatures upon infection. And in particular, this one gene really caught our eye. So PTPRC is a gene in a protein tyrosine phosphatase receptor family, and its protein product is mostly known as CD45. So the isoform variation of CD45 has been considered as a marker for functionally distinct immune cells, such as activated T cells or memory T cells. And upon the infection of SARS-CoV-2, we see that only in females, like this different bar, this is control first-time PCR and subsequent PCR. We see that only in females, the exon 6 inclusion level is being upregulated, versus in males, this inclusion level remains constant. Because exon 6 corresponds to the extracellular protein domain of CD45, this is functionally important and marks a distinct immune cell repertoire alteration that is restricted in the female cohort. We can also see a widespread sex differences on the transcriptional level. So for this purpose, we focus on a set of genes called interferon-stimulated genes. So in short, interferon-stimulated genes, or ISGs, are the genes that are triggered by the interferons and provides the host defense to fight off a wide range of pathogens. So we compiled a list of 361 ISGs and performed this gene set enrichment analysis by comparing during infection samples to the baselines. And indeed, we see that for both males and females, these ISGs are being significantly upregulated upon the SARS-CoV-2 infection. This is expected because those are the host defense genes that will fight off this virus. However, the extent of upregulation is much, much higher compared to the males. If you look at this enrichment score, and this p-value is highly significant. So we can also further summarize those 361 ISGs by a latent factor analysis, and we can group them into 4 reproducible latent variables that are reproducible across external cohorts. So the first one is type 1 ISGs that are the genes that are responsive or triggered by interferon alpha and beta. Type 2 ISGs are the genes that are triggered by interferon gamma. And the third one that is highly expressed in neutrophils, and then we denote them as neutrophil associated. So interestingly, you find that for those 4 reproducible latent variables, these are even higher before the infection. Remember that for this unique cohort, we have the molecular profilings before the subjects are infected. And comparing the healthy males to healthy females, we see that these antiviral ISGs are even higher at the baseline level before they're infected. So taking those 2 factors together, this really indicates that females have a stronger antiviral defense to start with, and this defense is even more upregulated upon infection. That might contribute to why females have a better outcome in COVID-19. So now I've told you different pieces, but we really want to wrap this up with a full picture. So to do this, we perform this causal mediation analysis. So I don't have the time to go through all the technical details, but briefly, causal mediation analysis is a statistical framework that leverages this potential outcome framework, and we can use it to analyze the mediation relationships between 3 variables. Here, those 3 variables in particular corresponds to those biological sex, the immunity pre-infection, and the molecular and clinical differences post-infection. And I've put a simplified version of this results summarized in this graph. And the real task we have done is much more complicated than this. But briefly, we were able to recapitulate some of these known relationships. For example, type 1 and type 2 ISGs are triggered by interferons. And if during infection you have a higher interferon, you're more likely to experience symptoms such as fever. So this also explains our observations earlier that females are more symptomatic. It's simply an indication that their immune systems are working. And this is the same if you get a vaccine. Aside from that, we found that the pre-infection interferon gamma triggered genes will actually reduce the viral load upon infection, but this is marginally significant, so we use this diagram to represent it. Finally, this CD45 splicing variation that is only seen in females is mediated through this higher neutrophil associated genes and interferon gamma, again, before infection. So here, although this graph is mainly focused on the gene expression levels of ISGs, we can argue that the splicing of CD45 is apparently also functionally important by marking this adaptive immunity, and they also exhibit this strong sex difference pattern. And indeed, in general, RNA splicing is a primary contributor that links genotype to phenotype variations. In this landmark study published in Science a few years ago, the authors performed a careful analysis to understand the sources of complex trait variations. And they concluded that for some of the traits, the contribution of mRNA gene expression level and splicing are comparable or even as larger from the splicing side. At the same time, how those splicing variations triggered by the immune response to fight off SARS-CoV-2 infection still remains elusive. So that wraps up the first part, which is the sex difference story. And next, we will perform a more detailed analysis of RNA splicing in this trial cohort. So we can functionally characterize those splicing events by plotting their molecular changes upon this infection. So here the x-axis are ordered pre-infection, first PCR positive, subsequent PCR positive, and post-infection. They're ordered longitudinally, and each line represents one particular splicing event. We can see that there's a large number of splicing variations being regulated upon this SARS-CoV-2 infection. This dashed line represents the average across all those different differential splicing events. And we see that the largest molecular difference is triggered when subjects turned first-time PCR positive. We can also analyze which of the genes are differentially spliced. And indeed, we see that the genes undergoing differential splicing are the ones involved in neutrophil degranulation, viral process, and some protein trafficking, which are all will contribute to the immune response. So taken all together, this demonstrates that splicing variation is a core component that is accompanied by the immune response, especially happen acute and fast, to help the host to fight against the virus infection. So since we have these non-trivial splicing variations, we asked to what extent we can generalize these splicing variations as biomarkers to help us predict which COVID-19 versus negative control. So here, what we did is we can compile a logistic regression based on those splicing markers and make it use it to predict the probability of infection. So 1 means you're infected and 0 means you're healthy control. And one particular subject really pops out. This subject X, which is a 19-year-old male, developed a SARS-CoV-2 specific antibodies at day 56. And the antibody response is fairly strong. Because of the presence of this SARS-CoV-2 specific antibody, this is a strong indication that this subject contracted the virus at some point. However, through this entire enrollment in the study for this subject, he never reported any symptoms, nor did he have any positive PCR test. Note that it's not only single PCR false negative test, but it's a series of PCR tests that is never positive. And by applying our splicing-based logistic regression model, we can accurately capture that at day 42, the probability of infection is really high. And the time frame between this infection to antibody development is 14 days. It again roughly corresponds to our understanding of antibody generation after the infection. So this really indicates that the splicing biomarkers are very robust in terms of capturing those PCR false negatives. So we're really encouraged by this. So we work with Fluidigm company to implement a subset of those splicing biomarkers on microfluidic devices, and then evaluated it in a second independent cohort. And just through this simple analysis of PCA, we can almost perfectly separate the COVID-19 patients from the negative subjects. So this really demonstrates that our splicing markers now are very accurate and also robust across different platforms because we developed those biomarkers from RNA-seq, and now they're included as probes in those microfluidic devices. This is part of a bigger project that is funded by the Department of Defense that towards developing deployable devices for fast and accurate diagnostics. And we are very excited about those results and actively exploring this in a combination of gene expression and DNA methylation in a small portable device that can be deployed, say, in train station airports for fast and accurate detection for a range of pathogens. So hopefully at this point, I've shown you that splicing is really a treasure trove of not only just robust biomarkers for diagnostics, but also they're functionally important. However, compared to gene expression, the study of splicing is still somewhat understudied, and this is for a good reason. So using RNA-seq, the statistical power for differential gene expression coding usually saturates at 25 million reads, versus for splicing in this genome biology paper 3 years ago, the authors compared a range of popular tools out there for analyzing splicing. And even at 120 million reads, we still don't see a clear saturation for the statistical power. So the over-reliance of splicing analysis on the sequencing depth is really a major issue in the field. So as an attempt to resolve this issue, I've developed this deep learning model called DARTS. So DARTS is a framework of two components. The first one is called Bayesian hypothesis testing, which is an Bayesian inference module. And the second module is a deep neural network that is the predictive module. So in the green box, we can use the Bayesian inference with a flat prior to compile a list of high confidence differential and unchanged exons. These exons will then be trained in a neural network based off their local sequence cis regulatory elements and also a set of transacting elements such as RBPs that capture the cellular context. So during application, this trained DARTS DNA model will be used to generate a prediction for each exon on totally independent data sets. And then these predictions will be used as an informative prior back to this Bayesian inference in order to augment any application of interest. So the key for DARTS to success apparently is the generation of this accurate informative prior. So to do that, we train DARTS on big data in the public domain, including all the shRNA knockouts in ENCODE and tissue-specific splicing profiles in roadmap. So by training DARTS on the combination of those two consortium, this data, which has more than 5 million differential splicing events, the DARTS generates pretty well across different cellular contexts and upon different perturbation of RNA binding proteins. So just as a demonstration of how DARTS can be used in practice, here we leverage the fact that ENCODE has 28 control samples of K562 and 24 samples of HAPG2. We can pull those replicates to form this ultra-deep RNA-seq resource in order to perform this cell type-specific alternative splicing analysis. So this is our gold standard. Then we can move on to perform an exhaustive pairwise comparison. This is 28 by 24 individual replicate analysis by using the dataset specific features. So the idea here is really just using this shallower sequencing depth in individual replicates and see how they can recapitulate what's happening when you put them all together. And for each of these pairwise comparison, we can run both the inference module with a flat prior and with a deep learning generated informative prior. And we see that incorporating the informative prior always improves the performance for this inference module in terms of recapitulating what's in the ultra-deep RNA-seq resource. And more interestingly, the best gain actually comes from the lowest sequencing coverage. So for example, here, if the pool if you're pulling two replicates that in some only have 50 million reads, then incorporating this informative prior will give you an 8% gain in AUPR. Versus if you already have 220 million reads, then probably nothing can be improved anymore. And this is at least in part resolve the over-reliance of splicing analysis on the RNA-seq coverage. That is through the generation and incorporation of informative prior in this deep learning and Bayesian analysis. So this is all very good, but I also want to underline the importance of neural network architectures. So neural architectures refers to a wide range of hyperparameters that exist in neural networks. For example, how many layers you have for the neural network, within each layer, how many neurons, and for each neuron, what kind of computation operation you want to impose on it, etc., etc. So here, just as an example, the red line is the DARS model that I trained, and then compared to the blue line, which is a much shallower architecture, we can see that this shallow architecture is just not capable of generalize very well across different cellular contexts, and the performance simply saturates very early on. And this architecture tuning is important, yet it's very laborious and becomes exponentially harder for convolutional neural networks simply because the CNNs will have a much more hyperparameters to tune. So to resolve this, this move gets us to our second topic of today, which is automated deep learning to design the neural architectures. So I implemented this automated machine learning method in a name called AMBER, or Automated Modeling for Biological Evidence-Based Research. But why do we really want to do that? I think the benefits comes in twofold. So for people who are generating the data and who might not be so familiar with deep learning, I think AMBER can significantly alleviate the burden of developing a deep learning model by automating this process. And even for experts who was trained and works on machine learning, AMBER can help them to explore a much larger space of possible models very quickly and in a purely data-driven way. In particular, the process of AMBER works like this. So we can first, initially the controller network, which is the master that generates at generating neural architectures, is totally on-train. And then we can sample some network architectures from the controller network, train the models with, and get the validation loss or validation accuracy from an independent data set. And then this validation accuracy will be fed as a reward signal to this controller network so that we can train this controller network to generate better and better neural architectures over time. So this process is iterated, is done iteratively so that in this reinforcement learning setup, so that the controller network will be able to generate better and better architectures over time. So the algorithms implemented in AMBER is efficient and purely data-driven, and as I will just show you in a second, it is capable of searching over 10 to the power 30 viable architectures in less than 72 GPU hours. So yeah, I skipped a few mathematical details here. If you are interested and we have time, we can go talk about that later. But for now, we can apply this model on a classical epigenetics marker prediction task, which is given a 1,000 base pair DNA sequence, want to present the absence or presence of around 900 epigenetic markers. This is also known as the deep C task. So before the search, we can randomly sample architectures from this large model space. And here, different computation operations are colored by different colors. We can see because it's uniformly sampling, there's a combination of different colors. And aside from the computation operations, we also learn those residual connections, which has been considered as a key to training deeper and more performant neural networks in the modern deep learning. So after training the controller network, what we get is this new model called AMBER-Seq. So compared to AMBER-based, AMBER-Seq really only likes to use 3 different computations, which is the pink, red, and green. And this has a surprising resemblance to the choices of computation operations that is selected in DeepSeek, the human expert model. So they just tends to use the same computation operations, but comparing those number of parameters, we are only using 1/4 of the total parameters in the expert model. But comparing the performance, we're actually doing much better than the both this randomly wired model and this expert model. So you might think that 3% of AUPRC improvement is not that much, but remember this is the average AUPRC across 900 individual tasks. So a uniform 3% improvement across 900 tasks is actually highly non-trivial. So here in this case, we can show that this automated model is accurate and it's beats the human expert model. So in our paper, we did a lot of benchmark of how this automated AMBER-Seq model can be useful in different use cases, such as variant effect prediction and in GWAS heritability analysis. But here, just as an example, I want to show this particular SNP that alters the binding of a transcription factor called SPI1. So the prediction, so here we include both AMBER-Seq, AMBER-based, DeepSeek, the 3 models we just talked about, and also another 3 popular methods for scoring variant effects, delta SVM, DeepBind, and Jasper, the motif scoring method. So for this variant effect, different methods tend to give a very different prediction, with only AMBER-Seq and DeepSeek predicts that the G allele will increase the binding compared to A allele for this SPI1 transcription factor, versus all the other methods predicts the binding in the other direction. So from an independent ChIP-seq dataset, we know that the G allele is predominantly bound over A allele. So in this case, AMBER-Seq and DeepSeek are the winners, with AMBER-Seq having a much larger magnitude of a prediction compared to DeepSeek. This demonstrates how this automatically generated model can be useful in variant effect prediction and fine mapping in real world cases. So AMBER is as a framework is publicly available, and I'm actively developing and maintaining it in different methodology and application directions. And I would really encourage everyone here who's interested to check it out, and hopefully can convert your own biochemical experiments into a quantitative deep learning model, and even better, can use it to interpret how genetic variations will alter the molecular phenotypes. To follow up on this AMBER as a platform, I will show you that AMBER is actually a reusable pipeline across biological domains. So what I just told you is this epigenetic regulatory marker prediction, the DeepSeek task. We can simply adapt this to the CRISPR-Cas9 editing outcome prediction. So the underlying biology for those two are vastly different, but from a machine learning perspective, they're highly similar, and AMBER will serve as this reusable pipeline that can be generalized across different biological domains. So this brings the last component of this talk, which is automated and variant-aware predictor for Cas9 editing outcomes. I hope you are not so tired, because this is very exciting. So a bit of background. I hope you all know that Cas9 is a transformative biotechnology that is capable of editing the genomes. So through this programmable guide RNA, Cas9 can be directed to anywhere, almost anywhere in the genome through this target sequence, and introduce the double-strand break at the target site. But until recently, people start to realize that the repair profile for this Cas9-induced double-strand break is actually reproducible and predictable from the local sequence context. So what I mean by editing outcomes is, for example, this is your target genomic sequence. If you introduce a cut here, you will get this 1 base pair insertion around 60% of the time. And if you do these experiments multiple times, you will always get this 60% of one base per insertion. And this is dependent on your local sequence context. And apparently we're not the first one to use machine learning to tackle this problem. There exist at least 3 methods that aims to predict the editing outcomes. They use boosting machines, multivariate logistic regressions, and artificial neural networks to predict these outcomes. And the common characteristic of all those 3 models is that they depend on feature engineering to derive informative features from those raw genomic sequences, and then they need to tune their models' hyperparameters before the model can be deployed. So we reason that with convolutional neural networks, we can totally bypass this feature engineering step because CNNs can learn useful features directly from the raw input. And we further couple that with neural architecture search implemented in AMBER, we can also bypass this model engineering, thus creating this end-to-end framework for quantum for Cas9 editing outcome predictions. And that's what we did. We created this quantum predictor, which is a fully automated state-of-the-art predictor for Cas9 editing outcomes. So quantum is trained on synthetic sequences, a large volume of synthetic sequences, and then tested on endogenous genomic sequences in human T cells. So compare those performance on the T cell editing outcome predictions with existing methods, we see that quantum was able to substantially outperform the ones that is designed by human experts. And sometimes the accuracy gain is more than 20%. So now we have this state-of-the-art, highly accurate predictor for CRISPR-Cas9. We want to see how inheritable genetic variations will alter the editing outcomes. So the reasoning behind this is really because the editing outcomes are dependent on the local sequence context. Then any genetic variations that will alter the sequence context is expected to alter the editing outcomes as well. So as a particular example, I will show you this pair of SNP and PAM147, which is a target site for Cas9 in this important gene FGFR3. So if you have the reference allele T, this target is actually not bad at all. It has a predicted of over 80% knockout rate by introducing this frame shift. However, if you have the alternative allele T, then this will significantly drag down your predicted frame shift by more than 20%. I guess more importantly, this allele is highly frequent in the African population, with around 30% of minor allele frequency in the African population. So this really means that for any editing primary cells for patients or subjects coming from this African population, this target site is probably not a good idea. And to do this we can also do this uniformly across the genome. And that's how we created the Quantum DB, which hosts the over 5 million guide RNA targets and around 100 million estimated variant effects for Cas9 editing outcome predictions. So finally, just to bring this a little bit in a more clinical relevant setting, we can extract this particular gene in our Quantum DB called PDCD1. So PDCD1 is an immune blockade gene, and that is being currently knocked out in an ongoing clinical trial to treat non-small cell cancer patients. So here we plot all possible guide RNA Cas9 targets across this gene, where each column is a PAM site, and each dot is a genetic variation that can potentially alter the editing outcomes for this PAM site. So although there's numerous potential targets to knock out this gene, after considering the existence of genetic variations, we can see that it introduces a substantial amount of variations. And in particular, I want to draw your attention to those 2 guide RNAs in orange boxes. So those 2 guide RNAs are the ones used in this clinical trial. And we see that at least for one of them, there exists a variant that can significantly alter the editing outcomes. And we argue that in order to design more applicable gene therapies induced by CRISPR-Cas9, we should consider the existence of such variations and find more variant resilient hem-sites. To wrap this up, I've told you how we performed the analysis on SARS-CoV-2 infection, which is on the sex difference story, and how splicing biomarkers can be used for accurate diagnostics on a small device. And in the second part, I told you this automated deep learning method implemented and applied to build models for epigenetics and also for CRISPR-Cas9 outcomes. For the Cas9 editing outcomes, it actually has a strong clinical implications. And now I'll move forward to the outcomes and future directions. So I think my current and past research has really started deep into a full stack of biomedicine. We have these deep learning models that can interpret these non-coding variant effects on genetic variations. And more importantly, we have the capability of editing the genomic variations. Those deep learning models has also been applied to understand complex molecular effects on both chromatin and RNA molecules, including gene regulation of transcriptional and post-transcriptional regulation. And those molecular phenotypes can be used as biomarkers to enable like portable device and personalized medicines with therapeutic potentials. So building on top of all those research, I'm interested in overall, like the overarching goal of my research is to bring these unbiased and data-driven quantitative discoveries to the full stack of biomedicine. And in particular, I've outlined the 3 specific outcomes that I'm interested in exploring in the immediate future. The first one is developing AutoML framework for interpretable biomedicine deep learning. So I guess interpretability is apparently very important for building human trustworthy deep learning models, especially in the context of biomedicine. But I believe I'm uniquely positioned to bring a different perspective to this question by engineering one or more of the components under this AutoML platform, so that we can strike the balance between predictive and also biologically interpretable models. This can also be generalized to multi-modal data integrations, such as language models and image analysis. And on the molecular side, I propose to extend my current research on CRISPR gene editing by a more complete spectrum of predicted editing outcomes, and also systematically analyze the variant effects, personalized variant effects, and especially in the context of risk predisposing variants. Also, current CRISPR-Cas9 research has really focused on the coding sequences by simply introducing this frame shift to the coding sequences. And we know that the vast majority of the human genome is non-coding, and with a lot of regulatory elements, I want to extend CRISPR-Cas9 to couple with those molecular traits as well. So finally, this is inspired by the study of SARS-CoV-2 and how exceptionally well the splicing biomarkers are. I want to systematically study how splicing as a post-transcriptional signature can crosstalk with the gene expression and perhaps other post-transcriptional effects on the RNA molecules in the broad context of infectious diseases, including bacteria and virus infections. And with that, I want to thank my postdoc advisor, Olga, and all the Troyskaya group members, my collaborator at Mount Sinai that helped collect and analyze the CHARM cohort datasets, and my PhD lab and collaborators at Children's Hospital of Philadelphia and UCLA. The funding for my postdoc research comes from the Simons Foundation. And I want to thank you for your listening. And happy to take any questions.
Thank you, Frank. That's a very exciting and clearly presented talk. Now we have time for questions. Anyone wants to start? You can unmute yourself or you can type in your questions.
Yeah, in terms of predicting the Cas9 CRISPR sites, do you find that there's variation from cell type or cell line to cell line? Do you find that there's more variation across ancestries? Is there any variation across that in this data that you can see?
Right now, we don't see such strong differences. This is mainly because the DNA repair system or pathway is highly stabilized across different tissues and cell types. And maybe the only exception is in stem cells. I think the repair system is just more efficient in stem cells. For example, just to be more specific, we trained our model on synthetic sequences in K562 and then applied them in primary human T cells, and it just worked like extremely well. So I think at least from what we see right now, a lot of tissue specificity is not very strong for the Cas9 prediction. But I would speculate that will be different for the stem cells, and yeah, with like a different repair, with different repair pathways and activities. And actually, I have a collaborator on CRISPR-Cas9 who is really a pioneer in this field. They developed this Repertoire-Seq. I think they posted it in bioRxiv, but basically they can interrogate the repair outcomes when you knock out certain repair enzymes. And if you have a preservation of those DNA repair enzymes or pathways, that can really change the editing outcomes. But given the stability across different cell lines, I think so far we haven't spotted any strong signals from there.
Mm-hmm.
Hey, Frank, that was a really great talk. I was curious, can you talk a little bit more about how the parameter search actually works? You said something about it being reinforcement learning.
Yeah, okay.
A little more detail, and also I'm curious how it relates to Bayesian optimization. Is it similar or completely different?
So right now it's not very, it's different, but there are some recent work that try to bring these two works together. But I guess I can quickly show you some intuitions behind it. Just let me find my backup slide. Here we go. Yeah. So in supervised machine learning, usually we just try to run this approximate a function that with parameters omega, but here we have some other hyperparameters called A or architecture. So the architecture and the omega is really this nested optimization, right? So you determine this architecture first, and then those omega is like within this architecture, the parameters are trained by gradients. And then this architecture can be further break down in the case of neural networks is in a layer by layer basis, right? So you have you can sample each layer as a token from A0, 1, 2 sequentially. The autoregressive nature for this is really important to consider. And here we are really just interested in learning this conditional probability, right? So for example, condition on the fact that the previous two layers are convolution layers, what's the probability of the third layer in order to generate a high performance architecture? So we can do that through this introduction of a recurrent neural network that predicts this conditional probability. And how do we optimize this theta? This is where the reinforcement learning comes into play, where we can optimize theta with respect to a reward on the validation accuracy. And this is just like policy gradient by multiplying the log likelihood of generating a particular architecture with their reward. So the total combined objective is that we want to find a high reward with a large likelihood. And yeah, this is how it works in a nutshell. Does that make sense?
Yeah, this is this is really helpful. So at a high level, maybe the intuition is that you can cut down on the exponential search space by looking at correlations between layers that work together to give the best performance.
Yeah, exactly. And that's where like this is different from Bayesian optimization, because I don't think the naive Bayesian optimization is just treating different tokens separately. However, the recent development reveals that you can actually encode this neural architecture through some, like for example, you can encode it with a graphical neural network. So then this will be encoded in a latent space, and then that latent space can be optimized by Bayesian optimization fairly efficiently. And actually, I think the method is called bananas, and this is kind of the state of the art. Right. Yeah, I guess I can extend briefly about this. So the benefit, the bonus from understanding the math is so the previous setup is really the basics. But if we understand this, we can further incorporate some of the biology descriptors for A0, right? So the vanilla form A0 is a marginal probability. It doesn't condition on anything, but we can condition on some biological descriptors so that we can study the interaction of biology with the neural architectures. For example, like ChIP-seq data sets tends to use this architecture, and versus DNA will use the other one. And now, aside from these simple tokens, we can also learn more complex structures such as residual connections, and we're actively exploring more like this biophysics, King-Altman diagrams type of stuff. So those are engineered from this existing mathematical framework.
Yeah. Can you hear me?
Right.
Yeah. I'm going to get back to what maybe also have been Liz's question. Can you go back to show the CRISPR that was used in humans where you had the good and the bad performing ones? Because if I understand this correctly, there are potentially variants in the neighborhood of where you're doing the CRISPRing. Now, the diagram where you said there's one in clinical trial that's currently being used. Was it CRISPR or was it antisense? I thought it was CRISPR. Yeah. So I think what Liz was asking, and if not, I'm asking now, is are the ones that are bad performing or predicting to be bad performing due to variants nearby that are not known? Are they more likely to be from genomes that are of non-Caucasian races, so that we select, when we make these clinical trials, genomes that are mostly Caucasian, but we may have a higher likelihood of getting into troubles, like in your example, when we are applying it to non-Caucasian samples?
Yeah, that's exactly what I'm trying to say. Yes. Right. So because the reference genome is supposedly derived from European ancestry, and I think there's a lot of other examples just like this. And I think for a clinical trial to be successful and widely applicable as a gene therapy, we should be able to find those target sites that are resilient across population and does not introduce other issues. Yeah.
Yeah, Frank, thanks a lot for the talk. We enjoyed it, and it really corresponds to our earlier conversations, and it's really a good basis for further discussions. I was fascinated by the sex difference stuff in the beginning between males and females in SARS-CoV-2 infection, and I was just fascinated by the fact that the males are suffering more, but when you looked at the symptomology and broke that down, the females were really showing off more symptoms and more categories than the males, and it was just driving me crazy to figure out what was going on there. I mean, and I saw that first line was symptomatic, and I was just wondering if you derived any insights in terms of what's going on there. I mean, it's almost as if, it's the silent killer. I mean, it's the males are suffering more, but the females are showing more symptoms, and I just couldn't put my head around it.
Yeah, that's definitely a valid concern, and I think it's due to the uniqueness of our cohort. So because they are all like marine recruits, they're physically fit, and as you can see, like over 50% of them are asymptomatic, and none of them need any of a hospitalization or anything severe. Like there's purely very mild symptoms of COVID if they have any. And on the other note is I think this has very interesting link to the vaccine side effects. So we know from various sources that taking the mRNA vaccines, females are more likely to experience side effects such as like a range of different side effects. I think it's just and from the causal mediation analysis that we performed, I think this can really be linked to the underlying immune systems and interferons. This is an indication that females have a very strong and active immune response that is actually working. And when someone's immune system is working, you will inevitably have some symptoms such as fever. And I think we have some wild speculations on this as well. I think this might be linked to the evolution of why females have a stronger immune system to start with. Yeah, as far as what our data suggests, I think that's the explanation.
Thank you. It's fascinating.
Yeah. Frank, I want to ask about the automated system, the AMBER. How much tuning is needed when you port it to different systems? I guess if the input data has a lot of, say, cell type stratification, you just name the stratifier, then you can walk away. It will do the job. Is that right?
Yeah, I think it's a fairly automated process. I think it's at the worst case, you can just specify a large enough model space and then run it to explore the best, like optimal architecture from this large model space. And given the efficiency of this search, you can run it multiple times and see the variance between the performance.
Say if the input data is not a linear sequence, but like Hi-C, it's a graph or 2D image, would you need to touch the architecture dramatically?
I would think that you need to compile the data, maybe a little bit efforts on that part. Can you hear me?
Yeah.
Because I think my internet just blinked a little bit. But yeah, what I'm saying is perhaps the most efforts will be compiling the data in a way that's machine readable and friendly. Yeah, but the underlying machine learning framework will be fairly similar if you are trying to train a convolutional neural network. And I mean, if you want to train a graphical neural network in the case of Hi-C, I think that's not something we can do right now, but we're definitely very interested in exploring how graph neural networks can be searched.
Yeah. Okay, we're at the end of the hour. Anyone wants to ask a last question? Okay, if not, well, Frank, thank you again. Yeah, for the wonderful talk.
Thank you for hosting this, and I really enjoyed it. Talking with everyone. Yeah.
Great. Okay, bye-bye, and everyone take care.
Bye.